Introduction. Medical nanotechnology is an emerging field that has the potential to revolutionize the diagnosis, treatment, and prevention of various diseases. It involves the design and development of nanoscale materials and devices to interact with biological systems at the molecular level. In this comprehensive script, we will delve into the various aspects of medical nanotechnology, with a specific focus on liposomes and Micelles as promising drug delivery systems. Liposomes and Micelles are lipid-based nanocarriers that can encapsulate both hydrophobic and hydrophilic drugs. The composition of lipids plays a crucial role in determining the stability and therapeutic efficacy of these drug delivery systems. The optimization of lipid composition involves carefully selecting the types of lipids, their concentration, and the ratio of each lipid in the formulation. Researchers have investigated various lipids, including phospholipids, cholesterol, and glycolipids, to enhance the stability, drug loading capacity, and release kinetics of liposomes and Micelles. Furthermore, modifications such as PE gelation and incorporation of ligands have been explored to improve targeting and reduce immunogenicity. One of the major challenges in treating neurological disorders is the inability of many drugs to traverse the blood-brain barrier, BBB. Polymeric Micelles have garnered significant attention as potential carriers for BBB delivery due to their small size, biocompatibility, and ability to encapsulate a wide range of drugs. These nanocarriers can be engineered to enhance BBB penetration through surface modifications, such as attaching targeting ligands that recognize receptors on brain endothelial cells or using stimuli-responsive polymers that can respond to specific triggers in the brain microenvironment. Such strategies hold great promise in improving drug delivery to the brain and treating neurodegenerative diseases. Prolonged circulation time is essential for improving the therapeutic efficacy of liposomes and Micelles. Polyethylene glycol, PEG, is commonly used to modify the surface of these nanocarriers through a process known as PE gelation. PE gelation imparts a protective layer onto the nanocarriers, reducing their recognition and elimination by the immune system. Various PE gelation strategies, such as covalent attachment, self-assembly, and mixed polymer systems, have been explored to optimize the circulation time, reduce immunogenicity, and enhance targetability of liposomes and Micelles. Targeted drug delivery aims to selectively deliver therapeutics to specific cells or tissues, minimizing off-target effects and improving drug efficacy. Ligand-conjugated liposomes are designed to recognize and bind to specific receptors overexpressed on the surface of target cells. These ligands can be peptides, antibodies, or small molecules, and their conjugation onto the surface of liposomes allows for precise targeting. Various receptor-ligand interactions have been explored, opening up possibilities for personalized medicine and improved treatment outcomes. Stimuli-responsive liposomes are designed to release their payload in response to specific cues in the microenvironment of the target site. This approach enables controlled drug release, minimizing systemic exposure and off-target effects. Different stimuli, such as changes in pH, temperature, light, or enzymatic activity, can be exploited to trigger drug release from liposomes. These responsive systems offer exciting opportunities in the field of drug delivery, allowing for on-demand and site-specific therapy delivery. Liposomes and Micelles can serve as attractive platforms for vaccine delivery and immunotherapy due to their ability to encapsulate antigens, adjuvants, and immunomodulators. Lipid-based vaccines have shown promise in enhancing immune responses against infectious diseases, cancer, and autoimmune disorders. Moreover, the design of lipid-based immunotherapies can facilitate modulating immune responses by targeting specific immune cells or delivering immune checkpoint inhibitors. This expanding field opens up new possibilities for effective prevention and treatment strategies. Gene therapy has emerged as a promising approach for treating genetic disorders and certain types of cancer. Liposomes and Micelles offer a versatile platform for efficient gene delivery, protecting the fragile nucleic acids and facilitating their entry into target cells. Various lipid-based formulations have been developed to overcome barriers such as cellular uptake, endosomal escape, and nuclear targeting for successful gene delivery. This field holds immense potential for personalized medicine and addressing unmet medical needs. Hybrid liposome-Micelle systems combine the advantages of both liposomes and Micelles to achieve enhanced drug loading capacity, stability, and modulation of release kinetics. By adopting a core shell structure, these hybrid systems can accommodate both hydrophobic and hydrophilic drugs, thus expanding the range of therapeutics that can be encapsulated. Hybrid systems can be engineered by combining various lipids and polymers, enabling precise control over drug release dynamics and improved therapeutic outcomes. Nanostructured lipid carriers, NLCs, have gained significant attention as nanocarriers specifically designed for lipophilic drugs. These carriers utilize a solid lipid matrix with incorporated liquid lipids, allowing for better drug solubility and enhanced stability. NLCs offer advantages such as sustained release, improved bioavailability, and reduced toxicity for lipophilic drugs. They have shown potential in a wide range of applications, including cancer chemotherapy, antifungal therapy, and treatment of skin diseases. Topical drug delivery offers localized therapy for various dermatological conditions, improving drug efficacy and reducing systemic side effects. Liposomes and Micelles have been explored as delivery systems for topical applications due to their ability to encapsulate hydrophilic and hydrophobic drugs. These nanocarriers can improve drug penetration into the skin, achieve sustained release, and protect fragile drugs from degradation. The field of topical drug delivery using liposomes and Micelles holds promise in the treatment of various skin disorders. Understanding the intracellular fate of liposomes and Micelles is crucial for optimizing drug delivery efficiency. Upon cellular uptake, these nanocarriers undergo intracellular trafficking, which involves endocytic uptake, escape from endosomes, and localization in the desired cellular compartments. By studying the pathways and kinetics of intracellular trafficking, researchers can develop strategies to enhance drug release within target cells and overcome subcellular barriers. Fine-tuning the intracellular fate of liposomes and Micelles holds great potential in improving drug delivery outcomes. While liposomes and Micelles have shown significant promise as drug delivery systems, their scale-up for clinical applications poses challenges. Various aspects, including scalability of manufacturing processes, reproducibility, and regulatory considerations, need to be addressed. Achieving consistent quality, ensuring stability, and optimizing large-scale production are essential for successful clinical translation. Overcoming these challenges is crucial to harness the full potential of liposomes and Micelles in improving patient outcomes. In conclusion, the field of medical nanotechnology holds immense promise for revolutionizing drug delivery strategies. Liposomes and Micelles, as versatile nanocarriers, offer exciting opportunities for targeted and controlled drug delivery. Through lipid composition optimization, PE-gylation, ligand conjugation, and stimuli-responsive design, researchers are developing innovative strategies to overcome therapeutic challenges. However, further research and addressing scale-up and manufacturing challenges are necessary for clinical translation. With continued advancements in medical nanotechnology, the future of personalized and targeted medicine looks promising.